Yeah, sure. Mutual respect. Okay, good evening everyone and welcome back. We are on the week two of Basics of Crop Breeding and Plant Biotechnology live session. And I am Prashasti Pandey, PMRF Fellow and PhD Scholar from Department of Botany, Banaras Hindu University. We'll just start the session in like next 5 minutes. We'll just wait for a few other people to join and then we'll start with the session. Okay, so let's start with week two. So yeah, we know for all those who have attended previous live session, we will go through some questions from previous year's assignments so that it will help you solve this year's assignments. And we will go through one question. I'll give you some time. You can try solving it. If you know the answer, you can write it in the chat box or you can speak it out loud. Then we will understand the concept behind that question and I'll tell you the answer of it. Okay, so let's start. Okay, so the first question is which of the following is not true about Mendel's law? The options are the law of segregation states that alleles separate during gamete formation. The law of independent assortment applies to very closely linked genes on same chromosome. Traits are inherited as discrete units, and the law of segregation is observed during meiosis 1. I'll give you let's understand the answer. So the question talks about Mendel's law of inheritance. So let's first understand that what are
Understand that
Mendel's
what are
law.
Mendel's law.
So
So,
okay, just give me a second.
okay,
So
so
basically,
basically,
Mendel's
Mendel's
laws
law
are the
are the
basic
basic
principles of
principles of
inheritance,
inheritance, and this was proposed by
Mendel.
Gregor Mendel.
And this
And this
explains that
explains
how
that how
traits are
traits are
basically
basically
passed
passed
from
from
parents to
parents to
offspring. Okay,
offsprings. Okay,
so
so
basically,
basically,
this
this
talks
talks
about
about
inheritance.
inheritance.
So
So
from
from
parents
parents
to the offsprings,
offsprings,
how
how
and
and
in
in
what
what
ways the
ways the
traits are
traits are
basically
basically
passed,
passed,
and
and
it that
that
passing of
passing
traits
of
has
traits has
certain laws
certain
which
laws
was
which was
defined by
defined by
Mendel.
Mendel.
So
So
basically,
basically,
Mendel
Mendel
gave us
gave us
three
three
laws.
laws.
First was
First was
law of
law of
dominance. Okay,
dominance.
so
Okay,
basically, law of
so basically,
dominance
law of
talks about
dominance
that
talks
when
about that
there
when
are
there are
contrasting
contrasting
when there is a
when there
pair
is a
of
pair of
contrasting traits,
contrasting
that is
traits,
one
that is
is a
one is a
dominant trait
dominant
and the
trait
other
and the
one is
other
a
one is a
recessive trait,
recessive
then
trait,
in that
then in
case
that case
one allele
one
which is
allele which is
dominant will
dominant will
express
express
itself
itself
and it will
and it will
mask the
mask the
recessive trait.
recessive trait.
Okay,
Okay,
so
so
in case
in
when there
case
are
when there
two
are
traits
two traits
available for
available for
any
any
two
two
types
types
or
or
basically
basically
two
two
contrasting
contrasting
traits are
traits are
available
available
and one
and
of
one
them
of
is
them is
dominant and the
dominant and
other
the
one is
other one is
recessive,
recessive,
only
only
dominant one
dominant
will
one will
express
express
itself
itself
and the
and the
recessive
recessive
one
one
will
will
get
get
masked.
masked.
This was
This
called
was
as
called as
law of
law of
dominance.
dominance.
The
The
second law
second
was
law
the
was
law
the
of
law of
segregation.
segregation.
So the
So
law of
the law of
segregation
segregation
talks
talks
about that
about
if there are two
that
alleles
if
of
there
a
are
gene,
two alleles of a
these two alleles will basically
gene,
separate or they will segregate during the gamete formation so that each gamete will carry only one allele. So
each
if
gamete
there
will
is a
carry only one
chromosome
allele.
which will
So
have two alleles, say
if there is
capital
a
R and
chromosome which
small
will
r,
have two alleles,
during
say
gamete
capital R and
formation,
small r,
the two
during
gametes
gamete
that will
formation,
form will
the
have
two gametes
one
that
will
will
have
form
capital R and the
one will
other will
have
have
capital R
small r. And these two gametes will have single alleles only and
and
this will
this will
separate
separate
or they
or
will
they will
segregate
segregate
during the
during the
gamete
gamete
formation and
formation,
this is
and this
called
is
as
called
the
as the
law of
law of
segregation, which is
segregation,
the
which is the
second law.
second law.
And the third law is law of
And the
independent
third law is
assortment.
law of
So law
independent
of independent assortment talks about that during this separation of gametes, which is like capital R and small r,
separation
when
of
it gets
gametes which
separated
are
into capital
separated
R and
into
small
capital
r,
R or small r.
this separation or assortment separation or assortment happen independently of each other during gamete formation. That is, the separation will not depend on anything, and especially they will not depend on each other. Okay, so this is called as law of independent assortment. So basically, Mendel gave these three laws. Now, as I explained, this is just the figures of the same explanation. That law of dominance will talk about a dominant allele which is capital R and the recessive allele which is small r, and the dominant allele will basically mask the recessive allele. Similarly, in law of segregation, so law of segregation is explained by two generations. One is F1 generation and the other one is F2 generation. When a plant has basically two traits, capital so this will basically help us to identify what in what form the segregation happens. So segregation happens in different ratios. In case of F1, the segregation will happen in 3:1 ratio, and in case of F2, the segregation will happen in 9:3:3. Okay, these are the basically the uh, offsprings or the generations having certain types of traits that will be formed will be in this form of ratio. Okay, so this one is law of segregation, and the third one was law of independent assortment are independent of each other, and different traits are inherited independently. They are not dependent on each other. So this was different law. So coming back to our independent assortment applies to very closely linked. So in this, the first option was the law of segregation states that alleles separate during different gamete formation. So this option is correct. That yes, it alleles will separate during gamete formation. That is the second Mendel's law of segregation. The third one was that traits are inherited as discrete units. Yes, this is also true that traits. This is the third law of independent assortment. The traits are segregated as discrete units and they are independent of each other. And the fourth option was that law of segregation is observed during meiosis one. Yes, during meiosis one, the first where basically the chromosomes will cross over and they will separate into different alleles. So it is observed during meiosis one. So the option B, that law of independent assortment applies to very closely linked genes. So the tricky thing here was very closely linked genes. So when on a chromosome, if the genes are very closely, if they are present very close to each other, or they are very tightly linked, in such cases, during crossing over, these two genes will not get separated. So they are not, basically they do not the chances of them getting crossed over is very less. That is why the law of independent assortment will not apply to very close or the genes which are linked together. So that is why your option B is incorrect, and the answer to this question is option B, that law of independent assortment applies to genes which are not closely present or not linked on the same chromosome. Okay, so I hope this was clear. Now let's move on to our next question. What is the expected phenotype ratio in a dihybrid cross for traits that exhibit complete dominance? Your options are 9 is to 3 is to 3 is to 1 is to 2 is to 1, 3 is to 1, and 12 is to 4.
Option
If
number
anyone knows,
A.
okay. Option A, 9 is to 3 is to 3 is to 1. Anyone else wants to try?
Yes, option A is correct. So first, let's understand what is a
Yes, option A
dihybrid
is
cross.
correct. So first let's
So
understand what is a
dihybrid
dihybrid
cross
cross.
basically
So
talks about a
dihybrid
genetic
cross
cross
basically
which will
talks about
involve
a genetic
two
cross
traits.
which will involve
So
two
when we
traits.
combine two
Okay.
traits
So
together,
when we
in that
combine
case,
two
it is
traits
called as
together, in
dihybrid
that
cross.
case
If
it is
we
called
are
as
talking about
dihybrid cross.
only
If
one
we are
trait,
talking
then it will be called as monohybrid cross.
then it will be called as
Okay.
monohybrid cross.
So when we
Okay.
talk about F1
So when
generation,
we talk
basically
about
F1
F1
generation is an
generation,
example
basically
of
F1
monohybrid
generation is
cross.
an example
When
of
we talk
monohybrid.
about F2 generation, it usually is a dihybrid cross, but it can also involve
Dihybrid
monohybrid if
cross,
we are
but it
considering
can
only
also
single
involve
trait.
monohybrid if we are
So this is the
considering only
example
single trait.
of
So
a
this is the
dihybrid cross.
example
So now
of
here
a
we
dihybrid
are
cross.
saying that
So now
parental
here
generation,
we are saying that parental
we are saying that
generation,
parental generation
we
will
are saying
have
that will
two
have
types of
two
parents are
types of
there. One is
parents are there.
dominant, which
One
is
is dominant, which is
capital R and
capital R and
capital
capital
Y,
Y,
and the other
capital
parent
Y,
is
and the other parent
the
is
recessive one, which is
the
small
recessive
r,
one, which is
small y,
small
small
r,
y.
and
So
small y,
in this
small y.
case,
So
the
in
gametes
this
which
case,
will be
the
produced in
gametes
F1
which will be
generation will
produced in
be
F1 generation will
capital
be
R, small
capital
r,
R,
capital
small
Y,
r,
and
capital
small
Y,
and small y.
and small
So
y.
this is the gamete
So
that will
this
be
is the
produced
gamete
in
that
the
will be
F1
produced in
generation.
the F1
Now, if
generation.
we go for
Now, if
F2
we go for
generation,
F2
there will
generation,
be
there will
four
be
gametes
four
that will be
gametes
produced
that will
from
be
this, which
produced
is
from this, which
capital
is
R, capital Y,
capital Y,
capital R,
capital
small
R,
y,
small y,
capital Y, and
capital Y, and small r,
small y.
small y.
These are the four
These are the
gametes
four
that will be
gametes that
produced
will be
in the
produced in the
F2 generation.
F2 generation.
Okay,
Okay,
and when we
and when
cross
we
this,
cross this,
so it is
so it
like
is
in this
like in this
example,
example,
capital Y,
capital R,
capital R,
capital R,
capital Y,
small r, and
small r.
small y,
These
small
four
r. These
gametes,
four
when
gametes,
they are
when
crossed
they
with
are
each other,
crossed with each
so
other,
they will
so
produce
they will produce
certain traits.
certain
Okay,
traits. Okay,
so in this
so
case,
in this case, whenever
wherever
and
the
wherever the
traits will
traits
have
will
a
have a
dominant character,
dominant
that is,
character, that is,
wherever
wherever
capital Y and
capital Y and
small
small
r
r
will be present. So
will
in
be
this
present. So in
example,
this
basically
example, basically
capital Y
capital Y
denotes
denotes
yellow seeds.
yellow seeds.
Okay,
Okay,
and R
and R
denotes
denotes
wrinkled or
wrinkled
round
or
seeds.
round seeds.
Okay, so
Okay,
capital
so capital
R is
R is
basically
basically
wrinkled or
wrinkled or
round,
round,
and capital
and capital yellow
yellow
is
or
yellow or
green.
green.
So
So
capital Y and
capital Y
small
and
y.
small y. Okay.
So whenever
So
there is a
whenever there is a
dominant trait, that
dominant
is
trait, that is
wherever
wherever
capital Y will
capital
be
Y will
present,
be present,
the seeds
the
will be
seeds will
of
be
yellow
of
color,
yellow
and
color,
wherever
and wherever
capital R
capital
will be
R will
present,
be
the
present,
seeds
the
will be of round color.
seeds will be of
So
color.
now if you
So now
see
if
in
you
this
see in
particular
this
example, it is
particular
having
example,
small y.
it is
Okay,
having
so
small
both
y.
the
Okay,
yellow
so both the
green trait
yellow
is in its
green
recessive
trait
form.
is in its
So when it
recessive
is in
form.
recessive
So when
form,
it is
the
in
seeds
recessive
will
form,
be green
the
color.
seeds
That is
will
why
be
this
green
particular
color.
seed is in green
That
color.
is why this particular seed is
Similarly,
in
in this
green
case
color.
also, it is
Similarly, in this
small y.
case
So
also,
the
it is
seeds are
small
in
y,
green
small y,
color.
so the
If
seeds
you
are in
look
green
here,
color.
it
If you
is the
look
same
here,
case. Here also it
it
is the
is the
same
same
case.
case. Here
Okay.
also it is the same
So
case. Okay.
wherever the
So
yellow trait,
wherever
that is
the yellow
small
trait,
y
that
will
is
be present,
small y will be present,
the
there
trait will be basically in
the
its
trait will be
recessive form,
basically in
and the
its
seed will
recessive
be
form,
of
and the seed will
green
be
color.
of
Now
green color.
similarly,
Now
wherever the trait
similarly,
is
wherever the trait is
smaller, the
small
seed
r,
here
the
will be
seed
in
here
its
will be in
wrinkled
its
form.
wrinkled form.
So in case of this
Okay.
example, the trait is smaller. Here also it is smaller. Here also it is smaller. So the
Here
seeds
also it is
are
smaller,
wrinkled.
here
So
also it
now
is
if you
smaller.
count,
So the seeds
so
are wrinkled. So
there will
now
be
if you
four
count,
possible types
so
of outcomes. One
there
is
will be
that
four possible
that the
types of
seeds are
outcomes.
yellow and
One will
round.
be
The
that
seeds
the
are
seeds are
green and
yellow
purple.
and round.
The seeds
The
are
seeds
yellow
are
and wrinkled,
green
and
and
the
round.
seeds are
The
green
seeds
and
are
wrinkled.
yellow
So if you count it in case of
seeds
yellow
are
and round, it
green
is
and
one,
wrinkled.
two,
So if you count it in case of yellow and
five,
round, it is
six,
one,
seven,
2,
eight,
3,
and
4,
9. So
5, 6,
from here it
7,
comes
8,
the
and
ratio
9.
as 9.
So here,
Now
from
talking
here it
about
comes,
green
the
and
ratio as
round, it
9.
is 1,
Now talking
2,
about
and
green
3.
and round,
So
it
the
is 1,
ratio is 3
2,
here.
and 3. So
Now
the ratio is
yellow
3
and
here.
wrinkled.
Now
So
yellow
yellow and
and
wrinkled is
wrinkled.
1,
So yellow
2,
and
and
wrinkled
3.
is 1,
Here the
2,
ratio
and
is
3.
3,
So
and then
here the
green and
ratio is
wrinkled
3,
is
and then
completely
green and wrinkled
recessive
is
trait,
completely
which is
recessive
just
trait,
1, which is
which
just
is
1. So
just
the
1,
ratio
which is
is
this one. So the ratio
1
is
here.
1
The
here.
ultimate
So the
ratio
ultimate
that comes
ratio
in a
that comes in
dihybrid
a
cross, which
dihybrid
involves two
cross, which
traits, is
involves
9 is
two
to 3
traitors,
is to
9
1.
is to 3 is to 1. Okay. I hope this was clear. So the option A is correct here, which is 9 is to 3 is to 3 is to 1. Okay, so moving on to our next question. Crossing over is most likely do not occur in genes that are linked and close to each other, far apart from each other, located on different chromosomes, and located on plastid genome. Option A.
Okay.
Okay.
So
So,
option
option
A.
A.
Anyone has
Anyone
any
has
other
any other
options?
options?
Okay, so let's solve this. So basically, what is
What
crossing
is
over?
crossing over?
Crossing over
Crossing over
basically
basically
happens
happens
between
between
homologous
homologous
chromosomes. We have
chromosomes.
solved
We
this
have
kind of
solved this kind
question
of
in
question
previous
in
session as
previous
well.
session as well.
Basically, crossing
Basically,
over
crossing
happens
over
in
happens in
homologous
homologous
chromosome during
chromosome during
packaging stage
packaging
of
stage of
prophase 1.
prophase 1.
So
So,
prophase 1
prophase 1
basically
basically
happens in
happens in
meiosis 1.
meiosis 1.
Okay.
Okay,
The
the
first
first
meiosis 1
meiosis 1
stage,
stage, the first
is
stage is
prophase 1,
prophase
which
1, which
contains
contains
five
five
different
different
stages,
stages,
and out
and
of
out
this
of
stage,
this
one
stage, one
stage is
stage is
packaging, and in
packeting, and
this
in this
packaging,
packeting,
basically the
basically the
crossing over
crossing over
takes
takes
place.
place.
So
So,
when
when
genes are,
genes are,
so in
so
case
in
of
case of
chromosomes,
chromosomes,
okay, so
okay, so
suppose if
suppose
these
if
are sister alleles,
alleles,
okay,
okay,
so if
so
these are sister
if
alleles
these are
and
sister
the
alleles and
chromosomes
the
are present
chromosomes
very
are
far
present
apart.
very far apart,
So in
so
this
in
case,
this
if
case, if
these will
these will
cross
cross
over,
over,
there are
there are
more
more
chances
chances
that
that
this
this
gene
gene
will get
will
crossed
get
over
crossed
and
over
this will not.
will not.
Okay,
Okay,
so that is
so
why
that is
when
why
genes
when
are far
genes are
apart,
far
there is
apart,
high
there is
chance that the crossing over
high
will
chance
take
that
place.
crossing over will
In
take place.
second case, when the
In
genes are
second
very
case, when
close,
the genes
so
are very
suppose in
close,
this
so
chromosome,
suppose in this
two genes
chromosome,
are present
two
very
genes are
close to each
present
other.
very close to
Okay,
each other.
so when
Okay,
the crossing
so
over
when the
happens
crossing
and when
over
this
happens
part
and
gets
when this
crossed over
part gets
here,
crossed
when
over
this
here
part gets crossed
and
over
this
here,
part gets
there
crossed
is
over here,
very less
there is
chance
very
that
less
these
chance
two
that
genes will
these two genes
basically
will
get
basically
crossed over.
get
So
crossed
in
over.
such
So
cases
in
where the
such
genes
cases
are
where
very close to
the
each
genes
other,
are very
the chances of crossing over between them is either very low or in most cases it is absent.
either very
Okay, so therefore,
okay, so therefore tightly linked genes usually do not show any crossing over and they are usually inherited together.
so the answer to this
So
question is crossing over most likely will not occur in genes which are linked and close to each other. So yes, option A
linked
was
and
correct for
close to
this.
each other. So yes, option A was correct for this.
Okay, so now moving on
Okay,
to
so
next
now
question.
moving on to next
Which
question.
RNA is
Which
involved in
RNA is
translation?
involved in
The options are
translation? The
rRNA,
options are
tRNA,
rRNA,
mRNA, all
tRNA,
of the above.
mRNA, and all of the above. All.
Okay,
Okay.
let's wait if anyone else wants to
Let's
answer.
wait if anyone else wants to answer.
Now
Okay,
let's try to
so now
solve this
let's try to
question.
solve this question. The question talks about three different types of RNAs basically. So one was mRNA, the other one is tRNA, and the third one is rRNA. Okay, so first let's talk about the function of these three RNAs. So mRNA is basically the genetic code from the DNA. The translation, when the translation happens, the translated form of our genetic code, which is DNA, gets converted in forms of codons to form mRNA. So mRNA is the basic genetic code that gets translated from DNA to rRNA, and that specific RNA which is formed from DNA is called as mRNA. The second one is rRNA, which is also called as ribosomal RNA. So ribosomal RNA forms the structure and the catalytic core of the ribosomes. Okay, and the third one is tRNA. So tRNA, or it is also called as transfer RNA. So it has the role to bring specific amino acids to the ribosomes during protein synthesis. Okay, so now how proteins, basically proteins are formed. The process of protein formation requires amino acids. Okay, so in case of amino acids, mRNA is the basic guide that will tell the ribosomal unit that what amino acid is supposed to be joined to form this particular protein. Now, how this is done is basically a codon is three unit structure like AUG, UGG, UUC. Okay, so these three specific sequences present on mRNA forms a single codon, and this codon translates to a specific amino acid. Okay, so when on mRNA, the opposite strand. So, for example, in case of UGG, the opposite strand is ACC. Okay, so opposite strand means of UGG will be ACC, and ACC is called as the anticodon. UGG here is the codon, and ACC is the anticodon. Now, in case of ACC, so basically tRNA will have the function to identify this ACC. So the specific tRNA which will have the anticodon of ACC will have a linked amino acid to it. Okay, so in this case, suppose ACC will code for tryptophan. Okay, so when the tRNA which identifies ACC comes and joins with this mRNA, there will be a link formed between the two amino acids which are next to each other, and that's how the protein structure is formed. Now, in this case, the next codon is UCC, opposite anticodon of which will be AG, which is AG, that is linked to amino acid phenylalanine. So when there will be a amino acid which is phenylalanine link, and another another amino acid to this protein chain will be formed, which will consider methionine, which will have methionine. The next will be TRP, and the next will be phenylalanine. Okay, so this mRNA chain will keep on moving. I mean, actually mRNA is still, this ribosomal unit keeps on sliding in this direction, and it will keep on adding anticodons to the codon present on the mRNA, and tRNA will keep on identifying those anticodons
and these amino acids will basically join together and they will form a structure of Okay,
structure of.
so this is the
Okay,
process of
so this is the
translation.
process of
So we
translation.
have the
So
in
we have the
translation
in
we have the
translation
use of
we have the
mRNA
use of mRNA
as the strand
as
which
the
produces the
strand which
codon and
produces the
helps in
codon
recognition of
recognition of
anticodons.
anticodons,
tRNA which will
tRNA which
bring the
will
amino
bring the
acids for
amino
the
acids for
protein
the
to get
protein to
formed,
get
and
formed, and
rRNA which will
rRNA
form the
which will form
structure
the
of the
structure of the
ribosomal unit,
ribosomal unit,
which
which makes
entire
this
process
entire
possible.
process possible.
Okay.
Okay,
So in our
so in our
question,
question, which
which
talks
talks
about
about
that which
which
RNA is
RNA is
involved
involved
in
in translation.
translation.
So
So
rRNA,
rRNA,
tRNA,
tRNA, and
mRNA,
mRNA,
all
all
three
three
are
are
involved
involved
in
in
translation.
translation.
So
So
the
the
option
option
D
D
will
will
be
be
correct
correct
here,
here,
all
all
of
of
them.
the above.
Okay. I
Okay,
hope this was
I hope
clear.
this was clear.
Let's move on to our
Let's move
next
on to our
question.
next question.
If a
If a
heterozygous tall
heterozygous
plant
tall
is
plant is
crossed with
crossed with
homozygous short
homozygous
plant,
short plant,
what
what
proportion of
proportion of
offsprings will be
offsprings
tall?
will be tall?
Options are
Options are
0%,
0%,
25%,
25%,
50%, and
50%, and
75%.
75%.
25%.
Okay. If
Okay, if
anybody else
anybody else
has
has
solved it,
solved
you
it,
can
you
tell
can
it.
tell it.
C option 50%.
Okay.
Okay.
Anyone
Anyone
else?
else?
Yeah, it should be 50%.
Okay.
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 Okay,
So
so
let's solve
let's
it.
solve
It
it.
is a
It is a
little bit
little
of
bit of
numerical kind
numerical
of
kind of
question.
question.
So
So
let's solve
let's
it.
solve it.
So the
So the
question
question
talks
talks
about a
about a
heterozygous
heterozygous
tall
tall
plant
plant
and a
and a
homozygous
homozygous
short
short
plant.
plant.
Okay.
Okay.
So
So
basically the
basically the
parents are
parents are
capital T
capital
and
T and
small
small
t
t
and
and
small t
small
and
t and
small t.
small t.
Okay.
Okay.
So they
So
are
they are
talking
talking
about
about
what
what
is
is
the
the
probability
probability
of
of
offsprings
offsprings
that
that
will
will
be
be
tall.
tall.
Now
Now
when we
when we
learned
learned
previously,
previously,
for
for
if
if
capital
capital
T is
T
the
is the
trait
trait
for
for
tall
tall
and
and
small
small
t is
t
the
is the
trait
trait
for
for
short.
short.
Okay.
Okay,
So
so
any
any
offspring
offspring
which
which
has
has
capital
capital
T
T
as a
as a
trait
trait
will
will
only
only
those
those
offsprings will
offsprings
be
will be
tall
tall
and the
and the
other
other
ones
ones
will
will
all be
all be
short.
short.
If
If
the
the
trait
trait
has
has
both
both
recessive,
recessive,
if
if
both the
both the
traits are
traits are
recessive,
recessive,
then in
then in
that
that
case
case
the
the
offsprings
offsprings
will
will
be
be
short.
short.
Okay.
Okay,
So now
so now
let's
let's
try to
try to
make a
make a
Punnett
Punnett
square of
square of
this,
this,
and we
and
will
we will
just
just
cross
cross
the
the
two
two
parents
parents
because
because
this is so this
this
will
will
be
be
basically a
basically a
monohybrid
monohybrid
cross
cross
because we
because
are
we are
talking
talking
about
about a
single
single
trait.
trait.
Okay.
Okay,
So
so
this is
this is
how the
how the
Punnett
Punnett
square will
square
be
will be
made,
made,
and
and
when we
when
will
we will
form the
form the
gametes,
gametes,
here the
here the
gamete will
gamete
be
will be
capital
capital
T,
T,
small
small
t,
t,
and
and
here the
here the
gametes will
gametes
be
will be
small t
small
and
t and
small
small
t.
t.
So we
So
will
we will
just
just
cross
cross
this
this
in the
in the
Punnett
Punnett
square.
square.
Okay.
Okay,
And in
and in
this,
this,
okay.
okay,
So the
so the
first
first
trait will
rate
be
will be
capital T,
capital T,
small
small
t,
t,
small
small
t,
t,
small
small
t,
t,
capital T,
capital T,
small
small
t,
t,
and
and
small
small
t,
t,
small
small
t.
t.
Now,
Now,
only
only
this
this
and
and
this
this
will
will
have
have
tall
tall
as
as
traits.
traits.
So
So
when we are
when we are
talking
talking
about
about
tall
tall
traits,
traits,
there are
there are
two,
two,
and
and
small t
small
and
t and
small
small
t
t
will
will
have
have
short as
short
trait.
as trait.
So
So
the
the
short will
short will
be
be
2.
2.
So
So
now
now
it is
it is
simple
simple
maths. If you
maths. If you can,
there are
there are
total
total
4
four
traits. If
traits.
you
If you
convert
convert,
do
do
like
like
small
small
maths on
maths on
this,
this,
it is
it
2
is
by
2 by
4, which
4,
is
which is
1 by
1 by
2,
2,
which
which
is
is
50%.
50%.
Okay.
Okay,
So the
so the
answer to
answer to
this
this
question
question
is
is
50%
50%
plants will be
plants will be
tall and
tall and
50%
50%
plants will
plants will
be
be
short.
short.
So
So
the
the
correct
correct
answer to
answer to
what
what
proportion of
proportion of
offsprings will
offsprings
be
will be
tall?
tall,
The
the
correct
correct
answer is
answer is
C,
C,
50%.
50%.
Okay, I
Okay,
hope
I
this
hope
was
this was
clear.
clear.
Okay, so
Okay,
now
so now
let's move
let's
on
move
to our
on to our
next
next
question.
question.
What is
What
the
is the
main
main
outcome of
outcome of
independent
independent
assortment
assortment
during
during
meiosis? The
meiosis? The
options are
options are
reduction in
reduction in
chromosome
chromosome
number,
number,
mixing of
mixing of
maternal and
maternal and
paternal
paternal
chromosomes,
chromosomes,
production of
production of
identical
identical
gametes, and
gametes, and
maintenance of
maintenance of
chromosome
chromosome
integrity.
integrity.
Option B.
Okay.
Okay.
Anyone
Anyone
else?
else? Okay, so someone also said option B. So let's solve this. So, first, let's talk about independent assortment. Uh, so, the question talks about what is the main outcome. So, during meiosis, basically the homologous chromosomes will pair or align randomly, and this will lead to new combination of maternal or paternal chromosomes, which is also called as crossing over. Okay, so as a result of this, there will be a new genetic variation that is being created. This was a very simple question. So, what the other options talked about that there will be reduction in chromosome numbers. So no, in case in due to meiosis there will be reduction of chromosome number, but not due to independent assortment. Independent assortment talks about the separation. Or basically the part of the crossing over, and that the two alleles will get segregated independently and they are not dependent on each other. So during meiosis, yes, the reduction of chromosome number will happen, but not during independent assortment. Independent assortment does not cause the reduction in chromosome number. Okay, the other option was production of identical gametes. No, the whole and sole purpose of independent assortment or the previous part, that is crossing over, is to create variation. It is exact opposite of it. This particular independent assortment creates variation, so it cannot produce identical gametes. And the last option was maintenance of chromosome integrity. No, this is not the main outcome of independent assortment. It has nothing to do with the chromosome integrity, so this option was also wrong. So the correct answer is yes, B, mixing of maternal and paternal chromosomes is the main outcome of independent assortment during meiosis. Very good. So let's go to our next question. Which stage of meiosis is characterized by the alignment of homologous chromosomes at the equator? This is also very easy. We have already solved this question once. Your options are prophase 1, metaphase 1, anaphase 1, and telophase 1. Somebody replied B. Anyone else? Okay, we have one more B. Okay, so everyone is saying B. Yes, this is, we have already solved this question in previous class, that there are the basically the meiosis is a special type of cell division that will produce four haploid gametes or four daughter cells from a diploid cell, and it has two major stages, which is called as meiosis 1 and the other one is meiosis 2. Okay, so meiosis 2, all stages of meiosis are divided into four major stages, that is prophase, metaphase, anaphase, and telophase. Okay, only in case of meiosis 1, prophase 1 is divided into five stages, that is leptotene, zygotene, pachytene, diplotin, and dikinesis. And after this all combines to form prophase 1, which is now continued by metaphase 1, anaphase 1, and telophase 1. And the second part of meiosis is prophase 2, metaphase 2, anaphase 2, and telophase 2, which results in four haploid daughter cells. Okay, so in all these meiosis parts, metaphase is the stage where the chromosome will align and come into the center for and prepare itself for separation. Okay, so yes, option B is correct. That meiosis is characterized by alignment of homologous chromosomes at equator at metaphase 1. Okay, so moving on to our next question. A test cross is used to determine the phenotypic ratio of a cross, the segregation pattern of alleles, the genotype of an individual with dominant phenotype, and whether traits are linked. Okay, somebody replied C. Okay, we have one more C. Okay, so let's solve this. Let's talk about what a test cross is. So, test cross is basically crossing of an individual with a dominant phenotype. Okay, so crossing of an individual with a dominant phenotype with a homozygous recessive. So these two things are important. That when you cross a dominant phenotype with a homozygous recessive individual, and this is done to determine the genotype of the dominant parents. So what do we mean by this? So uh, in the previous crosses that we have done, so let's talk about the trait, say tall. Okay, so when we are saying that there is a the parent, one of the parent is tall, so that means that tall parent could be capital T small t, or it could also be capital T and small t. Both this basically both this combination will result in this tall trait only. So now, how do we know that whether the parent has this particular chromosome type combination or this chromosome type of combination? So these questions become very important when we are trying to check the chromosomal abnormality in the child, and there are certain diseases which get carried forward due to genetic combinations. Okay, so it becomes very important for the doctors to basically know what is the chromosome type of the parents, whether they are homozygous or they are heterozygous, and that will determine whether that trait can be passed on to the child. So how it is done is that when a dominant phenotype is crossed with a recessive phenotype. Okay, so if we cross capital T small t with say small t, and if we cross both capital T with small t and small t, so this will result in different types of outcomes, and that will help us determine what was the combination of the parent. Okay, so this, because we don't know this is our question. We just know that it is a dominant trait, so we have one capital T in the phenotype. We don't know what is the other one. The other one could be a dominant allele, or it could be a recessive allele as well, but the trait is tall only. So when this kind of dominant phenotype is crossed with a homozygous recessive, now why homozygous recessive is important? Because if we take this also as heterozygous, it can result into anything, and we will not know that what was the missing allele that we are trying to find. So when this dominant phenotype is crossed with homozygous recessive trait, to identify what this allele was, this is called as a test cross. Okay, so of course, if you cross this, and there are two possibilities. So if the parent was capital T, and it is crossed with small t, then all the resulting offsprings will be capital T and small t. Okay, that means that all the offsprings will have the tall trait. So in that case, the possibility, if we are getting the outcome as tall traits, then the possibility of the other one is other allele will be a dominant allele, which is capital T, capital T. Now, if suppose the other parent is heterozygous, which is capital T and small t, and you cross it with the recessive trait, then in that case, the ratio will change. Okay, the ratio will change to 50% will be tall and 50% will be short. Okay, so if we are getting this as the ratio, then in that case the parent was a heterozygous dominant, that is capital T and small t. So to identify whether the parent is homozygous or heterozygous, the crossing over with the homozygous recessive is called as a test cross. Okay, so what will be the purpose of test cross is to identify a genotype, to confirm whether the parent is heterozygous or homozygous, and it is usually used in animal and crop breeding, where we definitely want to know that the parent is homozygous, because if we are producing an elite variety and if the parent is heterozygous, this can result to some offsprings being tall and the other ones being short. That means some offsprings will have the dominant trait and other ones will won't. Other ones won't. But now we are trying to grow the elite variety. So in that case, it becomes very important that the parent is of a homozygous type, so that all the resulting offsprings will have the dominant trait or the trait that we require. Okay, so this is how a test cross is done. So the option C is correct. The test cross is used to determine the genotype of an individual with a dominant phenotype. You cannot determine whether the traits are linked together with this. The segregation pattern of alleles is also not determined by this, and the phenotype ratio of a cross is also not determined. Okay, so option C is correct. Okay, so the next question is, if the genotype is capital A, capital B, small b, capital C, small c, how many different types of gametes can it produce if A, B, and C are independent genes? Okay, you can try attempting this one. This requires a small formula. If you know that, you can solve this. Okay, two students have replied B. Okay, so yes, almost every one of you has replied B. So now to solve this kind of questions, when you want to find out the independent genes or the number of different gametes that will be produced, the formula is basically 2 to the power n. Okay, where your n will be number of heterozygous pairs. Okay, so here it is important to know the heterozygosity. Okay, let's take two examples here. The question was talking about capital A, capital B, small b, capital C, and small c. And let's take the other example as A, capital B, small b, and capital C, small c. Okay, now just by using this formula, so here the so three traits are there in both examples. Okay, but the important word is heterozygous gene pair. So here the heterozygous gene pair is 1, 2, and 3. But in this case, there are only two heterozygous genes pair because this is not heterozygous, this is homozygous. Okay, so that won't count here. So in this case, it will be 2 to the power 3, and here it will be 2 to the power 2. So this answer will be 8, and here this answer will be 4. Okay, so the correct answer to this question is 8, which is 2 to the power 3. But I just took this example to show you that the importance of the word heterozygous. Why heterozygous gene pair is important. Whatever combinations given in your question, you need to identify the heterozygous pairs of that and then use this formula 2 to the power n, and only then you will get the correct answer. You cannot use 2 to the power 3 in this case. It will be wrong because there are only two heterozygous pairs. Okay, so yes, option B is correct. 8 is the right answer for everyone who have answered. Very good. Okay, moving on to our last question. Which of the following is true about incomplete dominance? The options are: the heterozygous phenotype is the same as the dominant phenotype. The heterozygous phenotype is an intermediate between two parental phenotypes. The dominant allele masks the effect of recessive allele, and it is observed only in animals. Okay, we have option B as answers from most of you. Okay, so let's talk about incomplete dominance here. So, incomplete dominance is basically a pattern of inheritance where neither of the allele is completely dominant. Okay, neither allele is completely dominant. That means that heterozygous individuals will show an intermediate phenotype. So, in previous examples, when we were talking about capital T as tall and small t as a short trait, and the resulting generation was capital T and small t, we said that this will result in the tall phenotypic trait. Okay, but there are certain phenotypes in certain you know certain organisms where this particular genetic combination will not result in the dominant trait. Instead, it will produce some trait which is basically a middle part of these two dominant traits. So there is an example which is called as Mirabilis jalapa, or it is also called as 4 o'clock plant. So in this plant, if the parents are of capital R, capital R chromosome combination, and the other one is small r. So this particular dominant trait is red, and small r is white. So the resulting offsprings will produce capital R and small r, and this trait in normal conditions should be red because R is the dominant phenotype. Okay, but that is not the situation. The resulting trait here is pink color, which is an intermediate color, which is combination of red and white combination, and results in the pink color flowers. Okay, the F1 generation will be pink in color. So this is called as incomplete dominance because the R gene was not completely able to mask the recessive gene, which is small r. So therefore, neither R got completely expressed nor the small r was completely masked. So the resulting color was pink color. So in such cases, the F2 generation, when we calculate the F2, in normal cases, the F2 generation ratio is 9 is to 3 is to 3 is to 1. Okay, but in such cases, in cases of incomplete dominance, the F2 phenotype ratio is resulted in 1 is to 2 is to 1, where your capital R, capital small r will produce red and white flower, but your capital R and small r will produce so, okay, we are talking about monohybrid cross. So in case of monohybrid cross, the normal ratio is 3 is to 1. And the small r and small r will basically produce pink color flowers. So here the ratio becomes 1 is to 2 is to 1. Okay, and the phenotypic ratio here will be 1 is to 2 is to 1. Okay, and this is the example for incomplete dominances. Your Mirabilis jarapa, snapdragon flower, and one is called as blue feathers flower. Okay, so these are the examples of incomplete dominance. So the correct answer to this question is B. The heterozygous phenotype is an intermediate between the two parental phenotypes. Okay. So I hope all the answers to the questions were clear. If there is any doubt, you can ask. Otherwise, we will end this session and we will meet in the next session, which is next week, Tuesday.